Okay, then we're going to wrap up our discussion of networks and pathways in systems biology by talking about some of the resources we can use to actually get access to network and pathway information. And there's so much out there that I can't really do an exhaustive coverage of what's available, but we can at least see some of the basic tools that are available to work with this, some of the places you can learn about already known networks and pathways, and do some discovery on your own with genes that might be of interest to you. So what I'm going to do is jump straight into looking at some of these online resources and start with one we looked at very briefly last week called the Gene Ontology. So the Gene Ontology, as I mentioned last time, is a collection of effectively organized terms, so an agreed upon set of vocabulary for referring to aspects of biological function. And this solves a very important problem in biological inquiry, which is that different groups working on the same thing may use different words to refer to a common concept. And while human beings can often deal very easily with different synonyms for the same concept or the same location in a cell or the same biological process, it's much harder with computers. With a computer, you really need to be precise about what you're referring to, and so it becomes very important to have a way of standardizing our terms for referring to concepts in biology. And this becomes very important when we're talking about systems biology, because we have to know if we're referring to a given system that other people are talking about the same system or different systems or related systems, subsystems, supersystems, and so forth. And the Gene Ontology is an attempt to deal with all of that by creating an organized ontological categorization. So sort of like a hierarchy, but with multiple chains of inference that I'll show you about. And to see how that works, what I'm going to do is just go to this Gene Ontology webpage and jump straight to the tools for accessing it, which are organized under something called the Amigo Browser. So we'll go to Amigo here. And Amigo is going to bring up a few different options that we have available for working with the Gene Ontology. And what I'm going to do to illustrate how this works is just start with a simple query. So I'm going to type in a kind of function that might be of interest to us, and we can see what the Gene Ontology has to tell us about that function. So I'm going to type in here specifically a sort of enzymatic function, so something called an Adenylate Cyclase activity. And we can see that there is a Go term associated with Adenylate Cyclase activity. So this is a Go ID. It says that there is an entry within the Go database associated with that term. And I'll just pick this, and that will take us to a page that's associated specifically with this term, Adenylate Cyclase activity. So Adenylate Cyclase activity is a kind of enzymatic activity, and that's annotated within the Gene Ontology through this categorization known as molecular function. So molecular function is one of the three standard terms that are in the Go ontology. I think they've added a fourth one recently. But basically, molecular function refers to enzymatic kinds of activities. There are biological processes which refer to kind of higher level activities of the sort we'll see shortly. And last time we had seen cellular components, so basically localizations within the cell, another way they organize their terms. And what we have here is a collection of synonyms related to those terms, and then various information that might be useful to us if we cared about what this term means. One thing that I think is useful for illustrating what the Gene Ontology is to pick this graph view here. And what the graph view is going to show us is what it actually means to have a term within an ontology like this. So let me scroll to the right here. We have a high-level category, which is molecular function, and then there are different kinds of molecular functions that are categorized within the ontology. So there are catalytic activities, that's one kind of molecular function, and we're looking at something that happens to be a kind of catalytic activity. So there would be a page within the Gene Ontology associated with catalytic activities where we could hear all about different catalytic activities and learn about subterms and superterms associated with that. One kind of catalytic activity is something called a lyase activity, and we can guess from that being shown here that this is relevant to this adenylate cyclase activity. And if we keep scrolling down, we can see that there is another kind of activity called the cyclase activity. And within the lyase activity, there's a subtype of lyase activity. And when we get all the way down to the bottom here, we can see that adenylate cyclase activity is both a kind of this sort of lyase activity and a kind of cyclase activity. So that's the sort of information an ontology would contain, this kind of hierarchical categorization of terms. And this is useful because often we're interested in working at different levels of abstraction. Sometimes we might be interested in cyclase activities in general, and we want the higher level term. Sometimes we're specifically interested in adenylate cyclase activity. This is also associated with other kinds of information. So this blue arrow here is telling us that adenylate cyclase activity is associated with a particular kind of biological process called the CAMP biosynthetic process. So CAMP, cyclic AMP, is an important signaling molecule in cells, and we can see that there is a kind of biological process associated with that, and that itself is an example of several different kinds of biological processes. It's a kind of CAMP metabolic process, it's a kind of cyclic purine nucleotide metabolic process, and so forth. And there's a huge ontology of terms associated with those different categorizations that gets us back to the high-level biological process, showing that that very specific kind of biological process at the bottom is an example of many different kinds of higher-level processes. So an example of a single organism process, a cellular process, a metabolic process, all of which are kinds of biological process. So all of this kind of information is contained in the gene ontology, and we'll see shortly why we get a lot of power out of having a categorization of terms like this, of having this kind of hierarchy. Before I move on, though, are there any questions about what we've seen so far? Okay, so one of the things we would often want to do with this, if we're interested in a particular activity, is learn about genes that are associated with that activity. So we're looking at a page devoted to adenylate cyclase activity. So let's pick associations here, and that's going to tell us about genes associated with that term, so that molecular function. There are a lot of them, so I'm going to limit things a bit. There are a lot of different kinds of filters you can pick, but I will pick a taxon filter, which basically that refers to the kind of organism that has this activity. Let me scroll down a little here. And I will limit it to Homo sapiens. That term there, and then it's going to limit it to genes in humans that are somehow associated with that activity. And we can see that there are quite a lot of different genes available here that are associated at one level or another with this kind of adenylate cyclase enzymatic activity. Each of these will have different levels of term associated with this. Some of these may be more specific kinds of activity that are instances of adenylate cyclase activity. Some might be associated exactly with that term, adenylate cyclase activity, but lots of genes associated with this process. We might also be interested in looking at things from the point of view of a gene, and you can do that as well. We can pick, let's say, this gene, GALR2, which we can see showed up in this search because it's associated with negative regulation of adenylate cyclase activity. We can pick that gene, and we can learn all about other processes that are associated with that gene in humans specifically. So we can see that this gene has a lot of Go terms associated with it. It has a cellular component term, so this is telling us that this is associated with a particular location. It's an integral component of the membrane, so this is a protein that is bound to a membrane within the cell. It's associated with a biological process, so neuron projection development, and it's associated with various other kinds of biological processes and enzymatic activities. So that is an example of the way we can learn from the point of view of a gene, and of course you can follow these kinds of chains of inference to manually put together these networks of interactions and associations. If you care to do that, more often the real power of this kind of thing comes because we can download all of this information if we want to. I'm demonstrating in the web browser because that's something I can demonstrate in class here, but you could download all of this information if you cared to and do some of the more sophisticated kinds of analyses we talked about in the last couple of classes. So if you needed prior probabilities to help your own network inferences, this would be a good way of finding priors, maybe suggesting that genes associated with a common process are more likely than others to be under common regulation, and that could help improve your ability to put together accurate networks, maybe mixing in your own data with some of this prior knowledge. Any questions about any of that? All right, so one way these kinds of things are increasingly used is relatively recently added to this Amigo hierarchy, or the Amigo tools. I'll just go to here, click on this and get back to the start page. So one way this is often used now is to take sets of genes you discover in various other ways and try to figure out if there are processes or functions that are associated with those genes. We saw one example in the last class of a way we might get a set of genes, or not last class, a week ago. So we had seen how you might take a set of gene expression data and try to find genes that are significantly different under different conditions. Very often the way we would follow up on that kind of information is once we've got a set of genes to start to ask, is this set of genes that showed up in our t-test on our expression results associated with some particularly interesting biological functions? So a way that can be done is to take the set of genes, and I'm just going to pick a set of genes that showed up high in that search last week on genes that significantly differentiate between lung tissue of smokers and non-smokers. I will put them into this term enrichment service. We have selected here biological process and Homo sapiens, and I'll click on this submit. And what this will do is start running a statistical analysis based on the Go ontology to try to figure out if that particular set of genes is enriched for particular biological processes. Now, sometimes these things can take a long time to run. This may not actually finish quickly enough for us to go through it right now, but I put this here to illustrate that this is one of the main ways people are using these things today, and it's one of the important parts of the way biological discovery is done today, working with these kinds of high throughput analyses. So you get a set of genes out of your expression analysis or out of association testing or whatever, and you want to know, does that set of genes have something in common? The statistics for doing that are pretty involved with these kinds of ontologies, so these kind of nested hierarchical categorizations, but these have been worked out for these various kinds of tools, and so we can run these analyses. So this may not finish for a few more minutes. I'm not going to wait for this to finish now, but before I move on, are there any questions about how we would use these kinds of things? Any questions at all about gene ontology or why we might want an ontology or what that's good for? Okay, well, then as I say, it's unpredictable how long this will take, so I'm just going to move on. Maybe we can revisit that a bit later in the lecture. So that's a tool that's nice if we're trying to do our own discoveries. We're trying to find categorizations that are overrepresented in a set of genes. But often what we will want to do is look at known sets of biological functions or known kinds of pathways or gene expression modules and so forth that we already know are of interest to us. And we want a way of exploring how those are related to one another. And as with many things, there's actually some really nice information integrated into the gQuery systems and the other NCBI resources. I'm going to type in a term related to a kind of biological process called Irby signaling. So Irby signaling is a particular process. We'll see in a moment what that is. And we can see that this turns up quite a lot of results. And we can find that under the chemicals categorization here, there is a set of results associated with what are called biosystems. So biosystems is NCBI's resource for networks and pathways, basically its systems biology resource. And we can click on this, and we can see the sorts of things that show up as hits to this particular term, Irby Signaling, in the Biosystems resource. So we get sets of pages associated with different kinds of Irby signaling or different annotations of this. One of the things that's kind of good and bad about this resource is that it's a meta-resource. So it's collecting information from many other network and pathway resources. So there is a fair amount of redundancy. So what we're seeing here are different pages that are more or less devoted to the same thing here, and sometimes we just need to pick the ones that are most interesting to us. I'll limit this to conserved biosystems, which is just going to cut it down a bit to ones that are found across organisms, but still we have a number of different annotations associated with this biosystem. We can see that there are Go terms associated with these different biosystems, so we could go back to the gene ontology, learn about all of that. But I'm going to just pick one of these, and we can see what is showing up here. And the one that we want to look at here for the moment is this one here, Irby Signaling Pathway. So this one is built from another data resource that we're going to be looking at a bit later. But basically, we can get some general information here about what an Irby signaling pathway is. And as we'll see in a moment, a lot of information about what is found specifically in terms of the networks and components of this pathway. So we'll just jump back for a minute here. It looks like our gene ontology result is done. And I just want to show you what a result of this kind of thing might look like. So we can see here lots of different gene ontology categorizations that have shown up as hits to this particular term. And what this is doing that would be hard to do for ourselves is look at these categorizations and figure out if our terms are statistically significant. So what we get here are E values and P values associated with these different categorizations. We can see that none of these is that strong of a hit, but we would probably say that maybe something less than 5 times 10 to the minus second would be at least weakly statistically significant. We can see we've got a couple of results that showed up there. So what's called a secondary metabolic process, a cellular aldehyde metabolic process. So these seem to be things that are significantly, or at least weakly significantly associated with smoking effects on lung tissue. And if we really cared about that question, we could try to dig more into what those meant and why these results might be showing up. Maybe put together a new study using these pathways to generate a set of candidate genes where we might be able to find weaker effects than our whole genome expression search is able to find. So any questions about any of that? Yeah.
What does the plus and minus column represent?
So the plus and minus, actually, I'm not 100% sure. It's maybe saying that this is whether it's a positive or a negative association. So if it's overrepresented or underrepresented, I'm actually not completely sure about that. Any other questions? Okay. Well, just going back then to the Biosystems database, I want to illustrate a few things about this resource. We've got this diagram here. I'll come back to that, which is actually showing us a visualization of the pathways. We can get a set of genes associated with these. And as is often the case with these integrated NCBI resources, we have a lot of nice links back to other sorts of information. So we could pick a gene, let's say the RAF1 gene, which seems to be associated with Irby signaling. That would take us to an Entree gene page for that gene, where we could access all the other information we would always get from an Entree gene page, SNPs associated with it, gene structure, all of that. And although we didn't go through it before, you can also get from these Entree gene pages gene-centric information on processes or pathways associated with this gene. So we could start looking at Irby signaling, find that this gene is relevant to that, and then come back and find all the other pathways that are also associated with this gene. I'm not going to dwell on that, just be aware that that's one reason it's useful to work with these integrated NCBI resources. We can also get various other kinds of information. We can learn about proteins associated with this, so what's produced by those genes, and we can learn about small kinds of networks. So they may involve genetic regulation, protein-protein interaction, metabolic networks. And we could learn all about this and integrate to databases associated with small molecules. So we could click on this, and this would take us to yet another resource known as the Pubchem Resource, which has information centered on small molecules like this. So we could learn in particular what this small molecule is. We could learn about all of the pathways and interactions associated with that small molecule, what structures are known that we might use if we were trying to do homology modeling, to maybe come up with a drug that would block binding of this molecule to whatever proteins bind to it, what systems and pathways are associated with it, and so forth. So a lot more information than I can go through today, but just be aware that all of these kinds of things are linked together, and I encourage you guys in recitation or on your own to go through and try some of these links. I don't have time to show you, and see the sorts of information available here. So any questions about any of that? Okay, so we go back here. One of the things I skipped over that is kind of interesting and relevant to what I want to talk about now is this diagram here. And the diagram here is too small to really make out the details, but this is interesting in part because it's showing us more specifically how this entire pathway, or all of the components to it, are linked together, what they're doing as a unified system. But it's also interesting because this is a diagram that was put together in a different data resource that I often find better than this biosystems one if you really want to dig into detail about a specific network or pathway. So in particular, this diagram is something put together by a resource called the KEGG Pathway Database. And the KEGG Pathway Database is arguably the best of the resources for really learning about how these things work. And we can click on this, this diagram to go to the KEGG database and learn about what KEGG has to say about this specific signaling pathway. So I'll blow this up a little. Here's a larger version of this. And it will have different versions of this. I'm going to limit it to the human version of this. So that was showing a kind of consensus among many organisms. If we take the human one, then we can see lots of the components that are involved in Irby signaling. So there's kind of a visual code to this that you would have to learn to read if you really want to understand what this is saying. But I'll just point out a few of the features here. So we can see these double gray bars here, and that represents the cell's plasma membrane. So what's to the left of this is outside the cell, what's to the right is inside the cell. And we see we have here in these green boxes sets of proteins, and some of them are sitting in the cell membrane. Some of them are outside, so these would be extracellular signals, maybe secreted by other cells that are interacting with these cell surface receptors, and then triggering these pathways of protein-protein interactions. Some of these steps in these pathways are denoted by this plus P. Can anyone figure out what that means? Yes, the plus P would mean phosphorylation. So these are signaling pathways. They may have some steps that are not strictly phosphorylation. They're just interactions of physical interactions between proteins. But what we can see here is something we can kind of explain as a chain of signaling in which we have an extracellular signal that maybe triggers a cell surface receptor, passes through a signaling pathway, in this case something known as the RAS-MEK-ERK signaling pathway, converges through the nuclear membranes, that's what's shown by this dashed line here, to interacting with DNA and promoting various functions that the cell would then activate through perhaps transcriptional regulation. So angiogenesis, adhesion, migration, invasion. So this happens to be a kind of signaling pathway that's very important in cancer contexts, and all of these different kinds of activities are things that are important in how tumors behave, so promoting proliferation or migration of cells and so forth. So that's why someone might care about this particular set of pathways. But that's more or less the visual code here, and you can look through this and you can see how all of this goes together, and you can also see other pathways that are associated with this pathway that may link through different molecules here. So are there any questions at this point? Right, so this is a kind of information that may be useful to us if we're trying to put together candidate gene sets. So maybe we are interested in studying tumor development, or what's called tumor progression, and we want to pick a small number of genes so we can do really high quality gene expression assays. This might be a hint that if we're interested in cell migration and invasion, maybe we want to look at these specific genes that lie along the pathways that converge on that kind of function. Often we would want to go into much more detail in looking at these things, and we can see some of the detail that's kind of hidden behind these diagrams by picking on one link here. And I will just click on the RAS link. So RAS is one particular protein that's important in this pathway. And we can start to get an idea of some of the complexity that is behind that link. So we can see here that there is a page devoted to RAS. And of course, in real practice, if you were writing computer programs for this, you would want to download all of this information, get it available in text files on your computer, and you could do all the things I'm doing manually on a much larger scale computationally. But manually, we can observe that we have this gene RAS. It's associated with quite a lot of other pathways. And we have various other information associated with this, such as, for example, what diseases are associated with RAS. There are other pages devoted to specific diseases as a kind of biological process. And we could click through these kinds of things and learn about, let's say, how RAS is associated with a different pathway called the MAPK, MAP kinase signaling pathway. We could get another page showing us where RAS sits in that pathway, how that's interacting with some of the same players as the Irby signaling pathways. These often intersect like that, and how different kinds of cellular signals interacting with different kinds of membrane receptors can end up triggering RAS-MEK signaling and converging on different sorts of functions and interacting with different other signaling networks. So that's giving you an idea, I hope, of some of the complexity that lies within these sorts of databases. And this, I would say, is I think probably the best resource for getting access to the details on these protein-protein interaction networks. So seeing more, maybe not everything that's known, but a lot of what's known in both a nicely human-readable format, or sort of human-readable, as well as getting all of the details in text files that you can download and use for your own automated analysis. This also tends to be a great resource for metabolic networks. So I just wanted to show a bit of an example of that. And to illustrate how that works, what I'm going to do is go to the KEGG top level page. So the URLs are on the slides for the lecture if you want that. We'll go to the KEGG pathway database, and we can see some of the other kinds of KEGG pages that are available. So quite a lot of these are these protein-protein interaction networks. There's a whole large categorization. I'm not sure how many of these there are at this point. But there are also quite a lot of these devoted to metabolic or reaction networks. We can actually even get a page of kind of the global overview map of metabolism in general, basically what is conserved about metabolism across living systems. And I know you can't see the details here, but if we look at this, we can kind of see here the global map of cellular metabolism. So if any of you have taken or are going to take biochemistry, this is basically biochemistry. Whatever you're going to learn, it's in there somewhere. This little circle here is the TCA cycle, and you could dig into this. You can click on the individual pieces, get down to individual molecules you care about, dig into whatever you want to know about as far as what your metabolites are, what your enzymes are, and what they are doing and how they're interacting with one another. Often we would want to start a little simpler here, so let me go back and pick a different page. I'm going to go to one that's devoted specifically to glycolysis and gluconeogenesis. Let me just bring that up here. And we can see a more focused KEGG metabolic network devoted specifically to glycolysis. So again, if any of you have taken biochemistry, you will probably recognize a lot of the players in this network. So we can see here, for example, L-lactate, pyruvate, oxaloacetate. Those are names you'll probably have to remember in biochemistry if you haven't taken it yet. but we can also see all this detail about how these are linked together, what reactions are getting you from one molecule to another. And this diagram, again, is basically just an organizing thing that is helping us identify much larger resources of information that are embedded within these databases. So, for example, we could click on pyruvate here, and we could learn all about pyruvate. We can get to a pyruvate-specific page. We can see that that is what pyruvate looks like. And we can see that pyruvate is associated with a large number of reactions. We'll return to that shortly. We can see that's associated with quite a few important pathways. It's kind of a key hub in many different metabolic pathways. And we could learn about, let's say, different enzymes that interact with pyruvate. We'll see about that shortly. And we could dig further into how these work by going into individual reactions that might be of interest when we're looking at pyruvate. So this is a page from the point of view of one reaction, showing us one of the kinds of enzymatic activity that acts on pyruvate. We could learn about the specific molecules here. Each of these would have its own KEGG page associated with it. And we could learn about some of the actors that are involved in working with pyruvate. We can learn that there is an enzyme that catalyzes this reaction. There is a code for this enzyme. So there is actually something like the ontology, kind of hierarchical categorization of enzymes, where you can dig down to whatever enzyme function you want. And it happens that that particular reaction is categorized by the enzyme 2.2.1.6. It's another way of encoding enzymatic functions, and we can learn all about what that enzymatic function is. We can learn what pathways it's involved in, and we can learn what genes in various different organisms implement that enzymatic function. So again, there is a ton of information here, much more than I can go through today, but all linked together and all accessible for automated analyses where maybe you want to download a complete metabolic pathway, all the actors, all the different players, and develop your own code to ask about maybe statistics, network statistics that might be interesting in telling us about how metabolic pathways differ between humans and bacteria or whatever. So, as with many of these resources, as I say, I can't tell you more than a tiny fraction of what's here, but I hope this gives you kind of a flavor for some of the complexity of information that's available in this sort of resource. So any questions about any of that? There are other kinds of networks contained in KEGG, so I'm just going to show briefly an example of that. So let me just go back here. Not really going to dwell on this, but in terms of some of the other sorts of information here, of course we have lots of metabolic networks, lots of PPI networks. I'm not going to go through all of them, but also just other sort of random things that might be of interest to us. So another kind of network is a cell differentiation lineage, and here we see a network devoted to the hematopoietic stem cell lineage, basically how blood cells are created by differentiation from initial stem cell populations. There are whole other resources devoted to this, other kinds of networks and pathways. There's a lot more here than we can go through, but again, I encourage you just play around with this and see what's there. So that's basically all I wanted to say about the KEGG resource, but I do want to show you about some of the other ones in the remaining time in class. Are there any questions about KEGG or about network and pathway resources in general before I move on? Okay, so KEGG has very good information on protein-protein interaction, that is PPI networks, very good information on reaction networks. It tends not to be as good for genetic regulatory networks. And as far as I'm aware, there isn't one resource that is the thing you would go to learn about genetic regulatory networks. But I want to show you a couple of other databases that have a bit more information on the genetics, although less broadly, so more focused on specific organisms. And one of these, just an example of what these might look like, is the Psych Database. So biopsych is not specifically genetic regulatory information. It also has lots of information on metabolic networks, but more information on how these intersect with genetic regulation. And we can see how this works by typing in a term here, and I will type in a metabolic term, glycolysis. So we've just seen glycolysis. I will search for that, and we can see the sorts of information that this would have on glycolysis. We can see that there are a number of pathways in this data resource that are associated specifically with glycolysis, and I will pick glycolysis 1, so this is one particular kind of glycolysis, and we can see that this has its own kind of diagram associated with glycolysis. We have different options for changing the amount of detail available in this diagram. I'll try adding a little more detail so we can see a bit more about what's going on, and that will show us both the molecules, the small molecules involved in glycolysis, and it's telling us something about the enzymes that are involved in different interactions in glycolysis. So as with the KEGG resource, we can click on any of these things, learn about particular interactors in this database, and I'll illustrate that by just going to, say, fructose 1,6-bisphosphate. We can click on this to learn about the role of a particular metabolite. And one of the reasons I'm doing this is to show how you can get to other kinds of information you wouldn't find within KEGG. So we can see that we have this particular sort of metabolite here. We have various kinds of pathways and reactions in which it's involved. We can pick one particular reaction of interest. So let's say this fructose degradation reaction. We can click on that, get to a page devoted to that kind of reaction. We can see the chemistry involved in that reaction. So what are the reactants? What are the products? And we can learn all about the different molecules that are involved in that particular process. So we can see, for example, that there is a particular enzyme associated with this. We can see, if I blow this up here a bit, that this has one of those enzyme classification numbers. So we could look up this enzyme in the enzyme hierarchy, learn that this is an example of this is categorization 2.7.1.56, which is a kind of enzymatic activity 2.7.1, and so forth. And we could learn all about that particular enzyme. And one of the nice things we can get out of this that we can't get from some of these other resources is at least a little information on genetic regulation involved with expression of that enzyme. So giving us some of those links we couldn't get so easily from KEGG, from the genetic regulation to the metabolic regulation. And one of the reasons I'm going through these things is just to illustrate the point I tried to make in the last lecture, that even though we have these different kinds of networks, and we often abstract that there is PPI networks and genetic regulatory networks and metabolic networks, really in some sense there's just one huge network. It's all linked together and really can't separate any one kind from any other. They're all interacting in various complicated ways. So, any questions about any of this? All right, so one other resource I wanted to show you, which has even better information about genetic regulation, but even more specific, is something called RegulonDB. So RegulonDB is a really nice network on tremendous detail on genetic regulation, but just for one organism, so just for E. coli K12. So these kinds of things are worked out for some organisms. We don't have resources like this, or at least this level of detail for everything we might care about. There are systematic studies underway to get this sort of information for human beings, let's say, to try to figure out in great detail what is going on with genetic regulation in humans. There's still a lot unknown about that, but we can see what the kind of ideal is by looking at some of the resources here. I'm going to type in an E. coli gene, so LAC-Z. It's a gene many of you are likely to encounter at one point or another in your studies. We can search for LAC-Z, and we can see some information on the LAC-Z gene and the LAC-Z operon. just like with biopsych, we can view this at different levels of detail. I'm going to 5' region of this gene. So basically what's involved in regulation of the LAC-Z gene. So each of these little boxes here is telling us about a known transcription factor binding site in the vicinity of the LAC-Z gene. So a known regulatory site where some protein is involved in regulating whether LAC-Z is transcribed. You can see there are actually multiple start sites, so you may get slightly different transcripts of it transcribed, depending on which regulatory interactions are present, so which regulators are present, and to what concentration. But basically, this is telling us that even in what we might think of as a fairly simple organism, like an E. coli, you can get quite complicated patterns of regulation. Any questions about this yet? All right, so that's kind of just the beginning of what we can learn about this. We can scroll down. We can see that there is various information about this gene. So Go categories again. Once you know what a Go category is, you'll start seeing these everywhere in these biological resources we look at. And if we scroll down a bit more, we can get to the operon. And we can learn all about things from the point of view of the operon and the transcriptional units associated with this. If I click on the operon here, we can see the LAC-Z operon. So we can see that LAC-Z is in a common operon with LAC-Y and LAC-A. We can see something about the various transcriptional regulatory sites associated with it. And we can see some information about the different transcripts. And if we scroll down here, look at those transcriptional units, we can see that there is tremendous detail on exactly what is going on in the promoter of each of these units. We can get the consensus E. coli K12 sequence for these units. We can find out exactly where the binding sites for these different regulatory elements are. And if we were interested in putting together all of this kind of information, we could download the examples of all of these different binding sites, pick a particular transcription factor of interest, like the CRP factor, find all the examples of binding sites that are known for that, download those to our own resources, and let's say build our own tool for discovering new examples of these binding sites. So can anyone tell me, how would you do that? Let's say we wanted to discover CRP binding sites in a relative of E. coli, like maybe find them in Salmonella. How could we use the E. coli data to put together a computer program for finding CRP binding sites in Salmonella?
Like generate a model that would like scan the genome of Salmonella to find similar.
Yeah, so we would want to put together some kind of model that could tell us if new pieces of sequence fit the pattern represented by these ones we know about. For a transcription factor binding site, it's usually a defined length, so relatively small region. You don't generally have insertions or deletions, so a position-specific scoring matrix would often be a good tool for that job. We could collect all of the examples of things bound by that protein in E. coli for all the different genes that have binding sites for that. We could get a multiple alignment, build a position-specific scoring matrix, and then we could use that to go into a new genome and scan for things that look like possible binding sites for that transcription factor in a closely related organism. So that's basically one way we could use this kind of information. If these were more flexible and we did know that there might be arbitrary length spacer regions, so insertions or deletions within these, what might be a more appropriate computational tool? What might be used instead of a PSSM? Yeah, an HMM, a hidden Markov model, would be a good choice if we knew that this wasn't a defined fixed number of positions. But a transcription factor binding site, usually a PSSM is the right thing to use there. So that's just an illustration of some of this information that's available. There's one last tool I wanted to mention that we can't actually go through in class, but it's worth knowing about. That's something called transfact. So I can't go through this because you need to set an account and log into this and get access to what's available in TransFact. So TransFact is basically a library of these position-specific scoring matrices. So they use the term PWM, position weight matrix, but it's the same thing. So if you want pre-built PSSMs for thousands of different transcription factors to make this analysis easier, TransFact is a place you can go to get that. This is actually, it's a commercial tool, so the full TransFact you actually have to pay money to access, but they also have a publicly accessible version that academic researchers can use. So that's often a tool you would want to use if you don't want to build your own PSSMs. So I just mentioned that as one thing that's available to us. So any questions about any of this?